Hi everyone. We just finished our cancer immunology lecture in the last lecture, and today we'll be talking about immunological applications. And this first part is going to be on diagnostics and research, or tools that we use for research and for diagnostics. And then after that, we'll talk about organ transplantation and therapeutics. But because this lecture in the past has been quite long, and I had some feedback saying, oh, we wish you'd broken it up. So I'm going to break up this first part into two parts as well. So let's get into it. In this lecture, or in the next two lectures, I hope you'll be able to describe the principles and uses, including the advantages and disadvantages of these different immunological techniques or applications. So, what are these applications? They can be antibody generation. That's usually where it all starts. A lot of these reactions are based on antibody and antigen recognition. Immunoprecipitation, agglutination reactions, antibody-based assays, affinity, antibody-antigen interactions, microscopy, immunofluorescence, and flow cytometry. So how we're going to break it up is that part one, which is what we're going to talk about in this recorded lecture, which is the antibody generation, the immunoprecipitation, agglutination reactions, ELISA, lateral flow, and Western blots. So those all might sound like nothing to you right now, but in a few slides, you'll be quite acquainted with them. In the next lecture, we'll talk about part two, which is ELISA spot, surface plasmon resonance, immunofluorescence imaging, and flow cytometry. So a lot of new terms, a lot of new techniques, but hopefully this will give you some exposure and idea of what can be done when you want to study immunology, or if you want to use immunological techniques to answer your questions. And take note that a lot of these methods are both clinical and for research as well, as in they can be used to diagnose a patient if they're used in a hospital, but they're also commonly used for research in research labs as well. So again, as I mentioned, a lot of the immunological techniques relies on the antibody-antigen reaction. At some point, they will need to use that because that's the way you're able to detect and recognize different molecules. So it goes back to this idea of the antibody having the variable parts in the arms, or the FABs, and this variable part gives you a huge amount of potential. So it can recognize really anything that the B cell has been exposed to, or as long as it's been activated to that particular antigen. So they're basically more or less, I mean, nothing's perfect, but it can match the particular conformation of that binding site, and then it can trigger off reactions or be manipulated to be labeled and visualized, and so on. Now, we talked about this type of antibody, and this is basically a generic Y-shaped antibody. Of course, we know there are different classes, and they have slightly different structures. And you also have, polymers of antibodies where they can form dimers or heteromers or pentamers. But then there's also, in terms of the recognition part, we can talk about antibodies being polyclonal or monoclonal antibodies. So these have different uses. So polyclonal antibodies means the antibodies are directed to a variety of different epitopes on the antigen that was used to immunize the animal, and basically the antibodies that we produce are polyclonal antibodies. That's the natural type of antibody that's produced. But monoclonal antibodies are special in that they are the product of a single stimulated B cell against a single epitope. Yeah. So the way they're produced is different, because one recognizes multiple epitopes and one recognizes a single epitope. Now the picture here, I think, gives some indication. So you've got basically the antigen, and it's multiple epitopes, and you've got some antibodies that recognize a specific epitope to these. And in the polyclonal version, you've got antibodies, a variety of antibodies being produced, recognizing different epitopes of that antigen. Whereas with the monoclonals, you see the antibodies produced are only recognizing this particular epitope. So they won't so they won't recognize these other epitopes on that antigen. So this means it's usually more specific. The monoclonals are more specific. They're more consistent as you make different batches, but it's going to be harder to develop. It's more expensive. It takes longer. It needs more antigen, and you'll usually use a monoclonal if you really want high specificity, and if you want to detect something quantitatively, because you know you're only detecting one epitope of that antigen. Whereas with polyclonals, they're less expensive to produce. Really, you just need to immunize the animal and then just harvest whatever antibodies that's made. So it doesn't take as long. You don't need as much antigen. And you can use multiple types of species to do this. Whereas with monoclonals, typically we only use mice. So the disadvantages is that you might have lot-to-lot variation because you don't know in that mix of antibodies how much is recognizing which epitope, and it's difficult for you to make consistent batch-to-batch variation. But when you use polyclonal antibodies, is if you just want to detect something qualitatively, if you want antibodies quickly, if you want something that's cheap, and if you need a high affinity, because obviously it's recognizing multiple epitopes, so you'll have a higher signal as well. So for the polyclonal antibody, really it's just about conducting a series of immunization and then harvesting the blood and separating the plasma and taking the serum. For a monoclonal, it's a bit more complicated. So if, let's say, you've gotten the antigen, which then you use to immunize the animal, you need to isolate the spleen cells and separate the plasma cells. Mix it with myeloma cells, which is what we call hybridization. So you've got a hybridoma of the plasma cell and the myeloma cell. And then you choose the clones specific to each of these epitopes. So you'll have basically 4 different types of monoclonal antibodies against one antigen, because they'll be recognizing the 4 different types of epitopes of the antigen. Whereas for the polyclonal, it's really just harvesting and you'll get antibodies all mixed up, recognizing different epitopes. Now, once you've got the antibodies, you can do a number of things, depending on whether or not they're polyclonals or monoclonals. The first thing, a very simple method, is called immunoprecipitation, and it's really as simple as mixing the antibodies and soluble antigen in a solution. And if the antigen is polyvalent, that means it has more than one antibody binding site per molecule, means it has multiple epitopes. It can bind multiple different antibodies. So in this case, you'll get basically a large link complex, and once it's large enough, it will actually fall out of solution, or it will precipitate. And so this can then be spun out, and this is actually a good way for you to pull out antigens using antibodies, or pull out antibodies using antigens. So one thing to note is the immunoprecipitation only can happen if the concentrations are somewhat equivalent. Too high or too low of one will not enable a precipitation line to occur. And there's also precipitations in agar, like the Octoloni method, and I will show you in the picture next. So this is what happens for immunoprecipitation in solution. So in the lab, if we had been able to do the practical, so you would have immunized the animals, and then you would have harvested the blood, or at least the lab technicians would have, and then when we go into the lab, you will be mixing the antigen used to immunize the rabbits or the chickens, and then you'll be titrating it over a series of dilutions, and then you'll see the appearance of a precipitation line if there was actually antibody-antigen complexing. But it's a bit tricky to figure out what is the concentration of the antibody and the antigen in each of these solutions. So sometimes you wouldn't get the right amount, and you wouldn't get a line precipitating. But if it did, whoa, just trying to move my face, yeah. So if it did meet in this sweet spot concentration, you'll have this cross-linking occur, where multiple antibodies will be binding to antigens, and then they'll kind of cross-link. Then you'll have that precipitation line. In agar, which is the Arterloni method, you've got basically these different wells. So you've got here A, B, C, D, E, and F. And you've got the viral antigens placed in the center of the well, and the serum samples from different people in each of the surrounding wells. So B and D are both negative. You don't see a line there forming, yeah? So it's kind of clear here because the people have not been infected with this virus, so they don't have the antiviral antibodies. But for all the others, for example, E, F, A, C, you can see a line forming between this well and the sample well, the patient sample well. So the antigen well and the antibody well. See an a precipitation line forming here, which means this patient or this person has antibodies against the virus. So that's a way you can diagnose whether or not a person has a particular viral infection. Another method we use antibodies for antibody-antigen reactions for is agglutination. So this occurs when cross-linking is between multivalent antibodies and multivalent bacterial or cellular antigens. So this will cause clumping. So the clumping will form between the cells with the antigens and the antibody molecules. So for example, you can have this kind of hemagglutination when you add antibodies with antigens on the red blood cell. And this is how we do the test for the blood type as well, I think. So you've got basically this hemagglutination where you have these wells 1 to 9 are the same amount of the SRBCs, so the red blood cells, and then a twofold dilution of the anti-SRBC serum. So you're diluting the antibodies across the wells. So this well, number 10, only has the SRBCs and no antibodies. So it actually settles into a solid button, which means it's a negative reaction. But where you have a high concentration of the antibodies or the anti-SRBC, for example, in the first well, obviously as you go further down, there's less and less antibodies. So in this one, the cross-linking of the SRBCs and the antibodies will form clumping, so it doesn't settle at the bottom of the well. It'll just be clumped in solution, and it'll be cloudy. So that gives you a positive reaction. So this 1, 2, 3 is a positive hemagglutination reaction. 4, 5, 6, 7, 8, 9, 10. It means there's too little antibodies to actually form agglutination in the wells number 4 and above, and 10 obviously has no antibodies. So you can also do this with bacterial and viral antigens. So just now, this one, the antigens are on the red blood cells, yeah? So this one's, let's say, bacterial or viral antigens. So they also have certain multivalent proteins that can react with the RBCs. And so if you want to see if a patient has antibodies to influenza virus, you add a serial dilution of the patient's anti-serum, and then virus, and then the RBCs. So if the hemagglutinin antibodies are present, they will attach to the hemagglutinin molecules on the virus, and it prevents those molecules from inducing hemagglutination. So normally, certain viruses like the flu virus can induce agglutination of RBCs in general. So if a person has antibodies against this virus, it will block the virus from binding to the red blood cells. So it won't cause hemagglutination if the person has antibodies. Same principle with bacteria, but you can see the pellet is made out of bacteria cross-linked by the antibacterial antibodies instead. So the agglutinin titer is the reciprocal of the greatest serum dilution that elicits a positive agglutination reaction. So let's look at this example for influenza, this agglutination assay. So we've got sample A, the titer is 1 to 8. This is the reciprocal of 1 to 8 because this is where you actually see the hemagglutination. Yeah, at 1 to once the antibody is diluted 1 to 8, then you see agglutination. So the titer is actually 128. Whereas for sample C, you can see that it starts forming around dilution 1 to 64. So the agglutinin titer is 64, so the reciprocal of the dilution. And in this case, sample B, there is no neutralizing antibody, so all of the RBCs are agglutinated or hemagglutinated by the virus. Right, so now we go into antibody assays based on molecule binding to solid phase supports. So just now we were talking about what's happening in solution or in an agar. But there's a lot of things that you can do if you're able to immobilize either the antigen or the antibody on a solid surface. Yeah? So this allows us to do things like read things on a microteeter plate or do microscope slides or beads, and allows for more volume of samples that can be processed in one go. So this is the high throughput element. So like just now, it's really just one sample at a time that you can test. This one you can test multiple samples. So some of the formats common for this type of assay is radioimmunoassay, which have now been replaced by ELISAs a lot, but you still need to use it for certain measurements with very low concentrations of the analyte. So enzyme-linked immunosorbent assays, so they're similar to RIAs, but they use antibodies or antigens conjugated to enzymes instead of radioisotopes. And then we'll talk about lateral flow immunochromatography, which are a simplified version of the ELISA, usually for rapid point-of-care diagnosis. So radioimmunoassays, they enable measurement of very low concentrations of proteins like hormones. And what they do is based on radioactively labeled antibody or antigen, you can use it to separate antigen-antibody complexes from unbound reagents. So it's usually what we call a competitive assay. If there is presence of the analyte, then you'll have a lower reading. But if there's no analyte, then you'll have a higher reading because it's been bound by the reagents. So the most common label is the iodine 125, which binds to exposed tyrosine residues. So here in this picture, you have the unlabeled cytokine outcompeting labeled form binding to the plate. So the amount of reactivity per well drop. And the amount of the cytokine in the experiment can then be measured by interpolation. So if you're trying to measure the cytokine is competing for the labeled, for the spot that would be occupied by the labeled isotope. So the lower the readout, the higher the concentration of the cytokine present in the sample. Alternatively, people use ELISAs, so instead of using radioisotope labeled antibodies or antigens, people use enzyme labeled antibodies or antigens, usually enzyme labeled antibodies. So these enzymes can be quite sensitive as well. They're safer, obviously because you're not playing with radioisotopes, and they're less expensive. And they can detect antibodies or antigens either qualitatively or semi-quantitatively using the optical density measurements. Or if you can construct a standard curve, you can also measure quantitatively, and you actually know the concentration in nanograms per mil or micrograms per mil. So how does an ELISA work? So the basis is that you've got these plates that have wells, right? So the wells usually are 96 wells per plate, and you can put the antigen on the surface of the well or the bottom of the well. So this is called adsorption. So the antigen is absorbed onto the well with a buffer that helps it stick, and then you'll add another buffer that blocks all surfaces that are not occupied by the antigen. So this is basically to prevent non-specific binding, because usually these wells are also treated so that they are very sticky. Then you add the antibody that is enzyme conjugated, and if the antibody does recognize this antigen, it will stick, and when you wash off the excess, the ones that stuck will remain. And then you can add the substrate. So remember, enzymes, what they do is they catalyze reactions when there is a substrate. So the enzyme will catalyze the substrate to color change, and then the color change will be visible, and you can also read it on a spectrophotometer. So the enzyme system used in an ELISA is usually either alkaline phosphatase, that's one type of enzyme. It cleaves the phosphate group from the p-nitrophenyl phosphate, or the NPPP, and then it will produce a yellow color which absorbs light at 460 nanometers. Or you can use horseradish peroxidase, it's fairly common. So this one converts different substrates like TMB, DAB, ABTS into colored products through their hydrogen peroxidase oxidation. So this one you can use a spectrophotometer that and the color depends on whether or not you stop the reaction. So with the TMB, when there's a positive reaction, you see this color changing to blue. And then if you add an acid, it will change to yellow. And this is a stopped reaction, so it doesn't keep developing further. Because if it develops further, sometimes it also becomes quite, the data becomes quite noisy. It becomes less accurate as well. So then you will read it in a machine, the spectrophotometer, and you'll read both the blue and the yellow and correct it. So that's the readout based on the enzymes, but there are different formats to ELISAs as well. So you can use ELISAs indirectly to measure an antibody specific to an antigen. You can use a sandwich ELISA. You can do a competitive ELISA, so similar to what we saw with the hormone RIA. Usually this is for very low concentrations, so much more sensitive. Besides using labeled antibodies, we can also use labeled biotin strep-avidin detection systems. So biotin is a water-soluble vitamin that binds to the bacterial protein strep-avidin. So it's kind of like antibody-antigen kind of reaction, but it's not an antibody. It's a vitamin. And so it actually allows for tetrametric binding as well. So you have 4 biotins that can bind to one strep-avidin. So this allows for signal amplification. So if you imagine each of this is labeled, so you'll get a higher signal from one detection reaction. Right, so let's look at the different formats. So all formats will need an antigen, a primary antibody or a biotin labeled antibody. So then it needs a secondary enzyme labeled antibody, or an enzyme labeled strep-avidin. So remember, you've got different antibodies, or you can have biotin and strep-avidin. So I'll give you an example. So let's say you have the antigen is, let's go into the different formats first. So with indirect ELISA, you've got the antigen coded on the well directly, and then you have the primary antibody added. Then it will bind to the antigen if it recognizes the antigen. And then you add a secondary antibody that is labeled with the enzyme E, and then that will produce a reaction when you add the substrate. So you have a color change there. With the sandwich ELISA, you have an antibody coded on the well, and then you add the antigen, and then you add an antibody that recognizes the antigen that is labeled with an enzyme, and then you add the substrate, and then there's a color change. So like a sandwich, the antigen is sandwiched by 2 antibodies, whereas this one, the antigen is straight on the well, and then the antibody piles up. With the competitive ELISA, as we mentioned earlier, there has to be it's actually competing for vacant spots. So you have to pre-incubate the antibody with the antigen first. so that if there is antibody or there is antigen present, then it will bind before you add it to the well, because the well is already antigen coated. So when you add it to the well, the ones that are vacant will be able to bind. The ones that are not vacant will not be able to bind. And then you add the enzyme conjugated secondary antibody, which will detect any antibody that's bound to the well, and then you'll get the color. So what this tells you is if there is more and more antibodies that are not bound, then there'll be more of them will be able to bind on the actual plate, which will give you a higher readout. But what that tells you is there's not a lot of the antigen or the analyte in the sample. But if conversely you have less antibodies able to bind here, it means it's all being competed earlier, or it's bound earlier with the antigen or the analyte present in that sample. Does that make sense? Yeah. Have another listen to that. So more analytes or antigens in this initial solution means less signal. Yeah. Now, I'll give you an example of what we mean by primary or secondary antibody. So let's say you have an antigen here, and let's say this is a TB antigen, and then I add here a primary antibody which comes from a human being. The primary antibodies are not labeled, so they're just, for example, my antibodies. So I add there, and then it binds to the antigen because maybe previously I have encountered the bacteria and I've developed immunity. I just never knew it. So it will stick to these antigens. And then there will be another antibody, a secondary antibody that is labeled with an enzyme, let's say HRP. So it's an HRP labeled anti-human IgG. Let's say this antibody here is my IgG. Yeah. So this anti-human IgG enzyme labeled antibody, HRP labeled antibody, will stick to my antibodies that are sticking to the antigen that are stuck on the well. So after the final wash, adding the substrate, there will be a positive reaction and there will be a color change. So I'll know, oh wow, I actually had TB once, I never knew, right? Conversely, in this format, you can have, for example, an antibody that recognizes the antigen. So usually it's a monoclonal. So let's say it's a mouse anti-ESAT6. So ESAT6 is a TB antigen. So when you add the ESAT6 protein into the well, it will stick and bind to the antibody, the anti-ESAT6 antibody that is also stuck on the well. And then we just add another antibody that also recognizes ESAT6, so a different anti-ESAT6 monoclonal that recognizes a different epitope that's labeled with an HRP. So then this will form a color when the substrate is added. Now, a tricky thing about this type of format is you need to make sure your antibodies are not competing for the same bind sites on the antigen, or you will not have anything sticking, right? So, the monoclonal pairs have to be correct, or there have to be binding different epitopes of that antigen. So this is a way you can detect antigen in a sample. Now, let's look at a more simplified version of the ELISA. I call it the second generation, or the point of care version. The ELISA you need to run in a lab, but lateral flow assays works the same way, but it can be run outside of a lab. So it's a paper-based test that gives you a yes or no. It's rarely quantitative. There are a few that are semi-quantitative, and usually they require an equipment to read out. But their benefit is that they're very cheap. They're usually very cheap. They can work very fast. You don't need to have a lot of technical skill. Think about a pregnancy test, yeah? Anybody can run it at home if they can follow the instructions. Just add the sample. The liquid will then go through capillary diffusion through the conjugate pad. It will rehydrate the gold conjugate, and then the analyte will interact with the conjugate, and then it will move to the membrane and be captured. So then that complex becomes immobilized and concentrated, and it will form a signal, usually in the form of a line. And you'll also have a control line where all the excess gold conjugate runs through to show that the test is complete, just to show that everything's gone up already. So in this case, if you don't have, for example, the antibody or the HCG, so this is a pregnancy test. So if you don't have the HCG hormone, then there will be no line on the results line. But if you're positive, then you'll have either a faint one or a much stronger one, depending on how much hormone you have in your urine. But this can just give you an indication. It doesn't actually tell you how much you have. That's why it's qualitative. All it does is tell you, yes, you are, or no, you're not going to have a baby. So how this looks like, up close, it's just if you imagine the well for the ELISA. So now instead of being in wells, it's on a nitrocellulose membrane. And instead of adding things sequentially and washing it off, everything is now just flowing through in one go. So it has to work in one go like that. And so we, instead of using enzyme labeled, you use gold labeled, because the gold will, when it's complex, it will form the color. So you add the sample on the sample pad here, and then it will rehydrate the membrane, and it will pick up all the antibodies conjugated with gold or fluorophore or whatever. And then if it can bind to the test line, which is also a line of antibodies, then it will then form a line there. But if not, it will just bind to the control line, which will capture any excess antibodies. So here it's showing there, if this is basically a positive result, because you've got the sample containing all these analytes that you want to detect. The analytes are here, and then the gold is there, and then the gold will be released, and you can see both the test and the control line of a valid test. This is a cross-section of a lateral flow strip, both on the side and from the top. It looks fairly simple, but there's a lot of things that can go wrong as well, so they need to make sure it's simple and it works really well. Alright, so that's ELISAs and lateral flows. So now we go into another method called Western blot. So Western blots are useful when you want to identify a protein in a complex protein mixture. So just now we're thinking about specific proteins or analytes that we're trying to detect. Is it present? Is it not? How much is it there? In Western blots, you can actually try and take out or separate what proteins are present, or however many there are. And that involves first separating them using polyacrylamide gel electrophoresis, or SDS page. And then you transfer the bands in the gel to a membrane, and then you incubate that membrane with different antibody solutions. And if there is, if the protein you're interested in is present, then you'll have a band show up when you visualize the membrane on a film. So you can use the same enzymes as you use for ELISAs as well, because this also uses the enzyme labeled antibodies for the reaction. So how this works, again, let me move my face. Yeesh. Move. Okay. So here we have the first step. So a Western blot has to start with first SDS page or a similar page that separates whatever proteins you have in a mixture. So you start with a mixture of proteins, yeah, and let's say you want to denature, you want to do SDS page. So you denature it in SDS, and then you add it to this SDS page gel. And then you run it. Run the electrophoresis in the tank, and as the current is being applied, the higher molecular weight proteins will migrate up to a point, and then the lower molecular weight proteins will migrate further. So that's the first separation. It's entirely based on the molecular weight of the proteins. Yeah. So there can be a few proteins at the similar molecular weight too. So that's something that we need to consider. And then once you've gotten that separated in the gel can be, you can actually apply an electric current to transfer all of the proteins in that gel onto a membrane. It's pretty neat. So it actually, just by virtue of the electric current, it pushes it out of the gel onto the membrane. So now the proteins are immobilized or stuck on this membrane. And then you can put these membranes into different, basically, baths of antibodies. And you can add the substrate to the after you've added the secondary antibody that is enzyme labeled. So it's still the same step, primary and then secondary, and then you can image the Western blot membrane, usually on a film. So it's just this grayscale or black and white. So these bands represent specific proteins, or at least specific proteins of a certain weight. This looks like they probably use a polyclonal, so that's why it's recognizing multiple bands. Right. So, yeah, this is probably a clear image of the Western blot aspect. So this is the SDS page. You've got the protein separated. Goes into a blotting tank to transfer from the gel to the membrane. And then you put it into basically this bath with antibodies or labeled antibodies. And what this does is, just being in a tube allows it to constantly be covered by the solution. And the tube's usually placed on a roller, so it's constantly rotating. It is quite nice because you don't have to use so much of the antibody solution, and you have enough to just keep covering that membrane. And after incubating it some time, could be an hour, a couple hours, could be overnight, and then you transfer the membrane. You develop first the membrane with the HRP, and then you transfer it to the film, and then you go and image the film, and then you'll see antigen bands on your film. Right, so that's basically the first part. These are all different. You can see the variations, how creative people have been in using antibodies and antigens to tell them different things. So in the next lecture, we'll continue with part 2, which is ELISA, surface plasma resonance, immunofluorescence imaging, flow cytometry, and then we'll do a little recap. All right, have a good week, guys. I won't give you a quiz yet. I will wait until we finish this second part. Bye-bye.